I audible? Yeah. Thank you very much for great introduction, and thank you very much organizers for giving this platform to share. Uh, okay. Yeah. So I'm going to share some of our recent works which we are doing in our lab. Mainly we are focusing on in silico approaches for identifying potential biomarkers and as well as phytochemicals for therapeutics in prostate cancer. So it's a part of CAPSI consortium work which we are working together on different aspects. So I'm going to share. So prostate cancer, as we all know, like all other cancers, it is also having lot of catching lot of importance these days as it is growing worldwide, and as well as in India, it has been growing. So the cases have been reported quite a lot, and that made us to look in and focus on what are the how it is how we can handle it. So in prostate cancer, to identify and as well as it to get in the androgen deprived therapy and resistant prostate cancer and PSA methods to identify, detect the prostate cancer were there, and you can here there will be normal, there is an enlargement in the prostate, so that actually is different. In the normal, you don't have the enlargement, but in prostate cancer, you have the enlargement at the urethra region. So there are different androgen receptor hormone gene regulation transcription will be regulated and upregulated and downregulated, and that is where the system has its role. So to do this, we try to, as a part of the consortium, there are genomic studies were conducted where we have taken the FFP blocks of the two and 4 samples, prostate cancer samples and benign samples, and then did the whole transcriptomic shortened sequencing and as well as the downstream analysis. This is done by Nidhi Shukla and as well as women as a part of the work. So I'm just sharing the previous work which has been conducted and thereon how we have moved forward. So the sample collection, RNA extraction, and as with Illumina sequencing, RNA sequencing, and then there's that mapping the alignment clip links and differential expression and filter go analysis was done. Oh, sorry. So so this is actually published, and the whole protocol was already published, and we have identified the LNC RNAs after the pipeline running, and then we try to find what are the LNC RNAs which have been identified in our samples. So all these LNC RNAs and also the PCA prostate cancer targeted proteins, we have tried to pull them together and try to find whether there is any possible interactions possible between the LNC RNAs and as well as the proteins. So the targeted proteins which we have screened about LNC RNAs 12 and then 13 prostate cancer targeted proteins, and then also the androgen targeted proteins we have considered in this work and did the protein molecular interaction studies, and then later on using the HDOC we try to do the interaction studies and try to find that what is the possible interactions with this and which are the interfacial residues where we can the binding is possible with these pathogenic proteins. And then we have done a lot of structural based studies to identify LNC RNAs with the proteins and where they are binding, both with the pathogenic protein, prostate cancer, and they helped us to go on. So again, we have done with the androgen receptor targeted proteins. We screened the PDB IDs of those, and then we have considered them for the LNC RNAs, 12 LNC RNAs to do the interaction studies and try to find what are the possible interaction residues which are interacting, and this helped us to make an overview of what are the interactions possible with LNC RNAs and androgen receptors prostate cancer. And we have found that the CSA RNA 10 and UN0094 and okay. So these are the common LNC RNAs which are interacting, and then we have the one in residue which is unique, and that is the SCA RNA 10, which is unique and which is common in all these interactions. So that is one thing which we were able to identify, that scRNA is interacting both in prostate cancer targeted proteins and as well as in the antigen receptors. So another area where we are working is screening the phytochemicals for lead molecules, where Gloriosa superba is a perennial creeper which is found in South India and also some of the northern parts of India, and it is a well cultivated field having a lot of medicinal value also in the cancer as well, but no studies were reported with respect to prostate cancer. So that made us to think that whether we can identify any novel molecules as secondary products from the Gloriosa superba and take as potential leads. So that made us to look into the literature, and we have identified colchicine, gloriosine, which are quite a lot of studies were done, but for the dimethyl colchicine and as well as dimethyl gloriosine, there are not much reports for. We have found some literature in Chinese studies, but not in Indian concept. So, sorry. So what we did is we have screened these pathogenic prostate cancer proteins and their IDs, and then took all these ligand molecules and as well as 5-fluorouracil, which is an anti-cancer drug, and to do the screening of the proteins with the ligand molecules using the open source software Autodoc. We have done the interaction studies and did the visualization where the residues are interacting, and as well as data molecular dynamic simulation to identify the stability of the complexes. So after doing the with all these proteins, we finally concluded with two of the one protein 3LMY interacting or having the least binding energy, which is more efficient in having the interactions compared to that of the 5-fluorouracil. And then also the colchicine, dimethyl colchicine is also having better binding. So these made us to look into the interactions and then try to see whether they are binding to the same location of to that of the 5-fluorouracil and then dimethyl gloriosine, but they are at different sites, and it is a randomized document. So that made us to go for the MD simulation. We have used COMES, And then we have done the interaction studies where the MD simulation has also showed us at 150 nanoseconds we have done, and in that we were able to find more stability for the dimethyl colchicine with 3L-MY protein and ID, and then we found that it has better stability than that of the 5-fluorouracil, which is green in here. And similarly, we have done for the dimethyl colchicine as well, and colchicine, and that made us to know what are the hydrogen bonds and how they are actually interacting, and how the stability is. We have calculated the gyration, and then with this we were able to identify that one of the LNC RNA which was screened in the earlier studies, which we have taken considered, has a possible scope for prognostic signature for PCA, and as well as androgen receptor biogenesis. So, and here we also found that because dimethyl colchicine and dimethyl fluorouracil, they are present around 10% in the actual plant. So it is also a good possible. There is a lot of scope and screening or isolating these compounds from the plant, and try to go for the in vitro studies. So with this, I also work with molecular imprinted polymers with biosensors. So that is where I have some areas where I am working on, and I thank all the students who have been working on different areas where I am interested in. And we are actually located in Amrita University, and it's in the coastal area of Kerala, and this is our department, and our chancellor is River Mata Amritanandamayi. And we have different various branches of Amrita University, and apart from that, I'm also part of BioClues, where we encourage career-ready women, and as well as women in science, where they move forward, and if there are any chances of doing any kind of review articles or publishing something, we always encourage them staying home. They can always do that. So apart from that, I'm also part of the Abir Bioscience Bioservice, which is also highly, it's an awareness program for cervical cancer. So I thank all the organizers, and as well as who are my strengths in moving forward in the research, especially Amritanand Mai, and our dean, CAPSI Consortium, Nirmal Lohia sir, Prashant, Barkha, Nidhi Shukla, and women who were part of this work, and Systems Genomics Lab, and family and friends. Thank you very much. I have a small grant for by developing biosensors, which is actually funded by BIRAC. Thank you.
Thank you, Dr. Renuka. I guess everyone is waiting for the evening cruise on the Ganga. How we are waiting for cruise on Ganga? Very few people